The next session was on disease pathogenesis and consequences of infection, and Dr. Diane Griffin will present the comments from that group.
Well, our group also had a great discussion with lots of good ideas. Oops, that was not. So they really wanted to focus on what the important issues were with respect to both the virus and the pathogenesis of infection. And the first order of business is to understand the virus. And understanding the virus, both the structure and how it may be different from other flaviviruses, and also the replication cycle and the effects on cells. So where it replicates, we don't know this. And encouraging research on primary human cells rather than tissue culture cells to understand better what the tropism is of the virus, and then what effects it has on those infected cells. Are the cells killed or not? Exactly what kinds of pathogenic or cytopathogenic effects there are. The next level, once you understand better the virus itself, is the pathogenesis of the virus and how it interacts with its host. And for this, we're going to need both animal models. That's our second point. But we're also going to need to study human disease in a much more depth than has been done yet. So for animal models, the order of the day right now is to try to develop or identify appropriate animal models. The models that are being used right now are mostly immunodeficient mice or non-human primates, both of which have problems associated with them. And so are there possibilities of other species that may be important, and to recognize that different hosts may be appropriate for different questions with respect to pathogenesis. And so the discussion of the advantages of guinea pigs or rats or ferrets, a large number of animals were discussed. And they may be different with respect to congenital infection and trying to model that versus just the basic pathogenesis of the infection. And so then once you have your great animal model, then the host response and the control and persistence is a major question. And where is a virus persistent? And the immune response is going to influence both persistence and clearance of the virus, which is the other side of persistence. What do we need to do to accomplish clearance? And if we don't, where is persistence occurring? Protective immunity is a very important question. It will be relevant to vaccine development, which we really didn't discuss, but you need to understand both what the role is of antibody and of various antibodies with different biologic properties, as well as the cellular immune response to protective immunity and also to clearance of infection. And then because of what we know about other flaviviruses, particularly dengue, understanding cross-reactivity amongst the flaviviruses and the potential of that cross-reactive immunity to enhance infection in the host. And then thirdly, understanding human disease. That's the pathogenesis we really want to understand. And to do that, you're going to need to be studying people where they are getting infected. And so really promoting and accelerating the ability to do human research in the field, collect samples, distribute samples, share samples, etc., is a critical need that was identified. And once you've been able to do the research or initiate the research in the countries where the infection is occurring, the questions that need to be asked are the timing and location of the virus infection, the immune response. They're similar to the ones you're asking in animal models, except now you know more specifically what the questions are that you're trying to answer in your animal models. And an additional question is the genetic susceptibility to different forms of the virus infection, and and particularly with respect to neurologic disease, because again, Bray syndrome is usually an autoimmune disease, and so is myelitis or post-infectious encephalomyelitis. So these may very well be certain people and genetic backgrounds that are particularly susceptible to those complications. And lastly, the relevance of persistence in particular sites. Semen is one example for sexual transmission, but also in blood or other tissues that may be relevant for transmission of the virus to other individuals. So that's our summary, and I'd be glad to answer any questions or have that expanded on by people that were in our group.
Any comments? Thank you.
Okay.